Hi everybody, Bismillah ar-Rahman ar-Rahim. Now we want to go further for the second lecture. We speak about classification of the fungi. And this lecture introduced by Dr. Nadir Musa Khalil of Zaid, Associated Professor and first consultant of medical microbiology. There are many ways or many hypotheses of classification of fungi from all to recently today. There are classification mainly according to laboratory diagnosis to morphological characterize mold and yeast in general, but there are many different shape of mold and there is many different shape of yeast. Others they classified according to the site of infection. Many also of mycologists classify this fungi according to the ways of reproduction. So now we want to speak about classification of fungi, and this classification of fungi it depends or firstly of binomial cultures that Carlos Linnaeus 1707 and 1778 have many categorical of the classify of fungi according to the kingdom. The kingdom fungi is fungi, and the subkingdom achloronata means without chlorophyll, so we call it achloronata. So it is a fungi is different from plant according to the recent classification. And we from the divisions that we should note and observe it that every part of division or subdivision and class should end by this suffix. We call it mycota: ascomycota, zygomycota, phycomycota, deuteromycota. So this is a division. Any suffix end by mycota is a division. So we can make the order of this according to the part of the division. So we have also subdivisions we called mycotania. Any part of the class we call it mycotena. Mycotena, that means ascomycotena, basidomycotena, ascomycotena, and phycomycotena. This according to the end suffix that is subdivision. So we have a class, and these classes end by mycetes. Mycetes is the pronouns of the end of the fungi. So any word of like isomycetes, ascomycetes, basidiomycetes, phycomycetes, that means is the class. So we go furthest to mycidity, that means is any part of many type of subclasses. If we find submycidity, that means it's a class of. And the order we call it mycetaceae, mycetalis, we have Mucorales. This is the part of classification of order of Phycomycetes, and also the family Mycetaceae. That means any word end by Mycetaceae. This is a part of the family, and we have a genus. Always a genus should be in capital letter, and the species should be in a small letter, and the genus and the species should be written as a Latin name in italic. That means you use in a computer system the letters I, that it help you to understand exactly what we have about this part. And so we have morphological fungi, this the vegetative structure. There are three types of the fungi. We have what we call monomorphic. This is the big classification. That means the fungi could take one shape. If it is a mold or if it is a yeast, we call it monomorphic. If it have one shape, we call it monomorphic. If it is a mold or yeast. But if the microorganisms change it from yeast to mold, this we call this fungi, it is dimorphic fungi. So we have two type or three type of the fungi. We have only mold or yeast. We call this part is monomorphic. But if dimorphic fungi and dimorphic fungi classify, that means due to the change of nutrition or temperature, the fungi change it from yeast to mold, because if we change it the culture, because most of the yeast find in the tissue of a human body as a yeast form. But when we isolated this microorganism, and if we do not find suitable culture similar like brain heart infusion or blood agar, so the microorganisms change their morphology to the mold form. So this microorganism of dimorphic, it change depend on and nutrition, and also depend on what we call depend on nutrition or temperature. It came from mold to yeast form. And we have a mold. If we want to describe the mold, mold as a definition is a cellus, that means colony of mold or fleshy fungus, consists of long filamentous, that means many multi yeast of cell joined together, and this filamentous called hyphae, singular is a hypha. So the simple unit of the mold is hypha. Many of hypha we call it hyphae, and many of a hyphae we call it mycelium. In a mold, the hyphae contain cross wall. If the hyphae have septations, that's singular word, we call it septum. That means half line inside, and there is no separated. They are many separated cell, separated by cell wall or septum. We divide into mold. We call it septated hyphae, and also other hyphae. That means we call it concentric hyphae. That means diffusion of nucleus through all the cell wall. That means it's aseptated hyphae. So we speak now about septated hyphae, have cross wall dividing the hyphae into unucleated cell-like unit, or aseptated hyphae, concentric hyphae in a few classes of fungi. However, the hyphae contain no septa and appear as continuous long cell with many nucleoli, which we call concentric hyphae. And this part, if there is a damage or a disease of the fungi, all the fungi can dead. It is different from septated hyphae, which are can separated the fungi from each other. Septated hyphae they are these hyphae found in many pathogenic fungi, and it is very important for identification in direct microscope. It is elongated in shape with septated branches, terminal or lateral hyphae, and there are many types of the hyphae. We have many types, especially of imperfect fungi. We call it the deuteromycetes group, and dermatophytes species, which infected the skin from the head until your toe. And there are many types when we grow and isolated this fungi. We have many different types of the shape, included antennera, racquet, hyphae, spiral, and most of these dermatophyte type of the hyphae, most of them are hyaline. Hyaline means it is transparent. It is different from the meticulous, because we have a hyphae, the meticulous, that are very dark, that you cannot see the nucleus inside the cell. We have cytoplasmic granule hyphae, bactinated. Also, we have another. All this is a part of the shape: antennella, racquet, spiral, cytoplasmic granule, bactinated, reflexive. And part of these are hyaline. These are transparent, and the other part is, we call it the meticulous. That means are very dark. As you see, this electronic scarring of a hyphae, vegetative hyphae, and we see, as you see, septated hyphae branches in this picture. And we also septated hyphae, antellar hyphae carried freely branching antellar like in appearance like Trichophytum schollini, which lead to permanent alopecia in our hair if the person do not treat it when he is a child. Spiral hyphae always are coiled or screwed like Trichophytum mentigarophyte. These are antennera hyphae, as you see in this part of the picture, and these are what we call spiral hyphae, as you see, or screw crook or coiled. This cytoplasmic granuloma, and these are reflexive hyphae in this part at the end. If you see in the last picture of the end, and as you see, we have comb hyphae. We are many different types, and this reflexive hyphae. As this septate hyphae, rocket hyphae are often similar like rocket, which we play with this part. Enlarged, showing a swelling area resembling tennis racket. As example of this is Microsporum odwini, and also we have pectinated hyphae showing comb-like protection Microsporum reveal, and we have reflexive hyphae is a branching on a hyphae composed of short segmented trichophytum. As example, Trichophytum sudanensis make reflexive of branches of the hyphae in other opposite side. Also, we have a septated hyphae, as we say, hyaline, that means transparent. We could see seption, and seption is like acraminium. The meticulous showing darking, pigmented mold as alternaria. We have sterile, distorted, swollen hyphae, viscose cytoplasmic granules, as we see in the previous picture, that are very large. As example, we have Trichophyton violicium. As you see that we have many different type of septated hyphae, and this part in different type of the kind of fungi and responsible for many pathogenic, because sometimes the hyphae have the role of pathogen to involve in this part. We have septated hyphae mold, like as we said, depend on their color. Hyaline hyphae, dimeticulous. Hyaline hyphae include dimorphic mold, include dermatophyte, which are dimorphic, which are very fatal fungi and very pathogenic to our human, and did not any predisposing factor for causing a part of the disease. We have dermatophyte, and this part is very essential and lead to many parts of the disease. We had conidiophores. Conidiophore, it is a ways of reproduction that terminating in a swollen vesicle, and conidiophore branching into penicillus. Also, we have conidior. All these parts in the group C, these ways of reproduction, we will go further in the part of the lecture about the reproduction. We'll speak in details about conidiophore, the method, and how this group belong to what, and how this group it can make reproduction. Conidiophore terminating in swollen vesicles, and if it gets very special type of vesicles like Spraguelas. We have also conidiophore branching into penicillus like penicillium. We have conidia, it can be produced in a cluster, and conidia born singly as one, and this lead to differentiated between different group according to conidiophore. Dermatophyte include three types of the Gerna, like Microsporum, which include more than 70 species. We have Trichophytum, it have more than 21 species. Epidermatophytum species, which included two important medical species, and dermatophytes as anembertic fungi. This conidiophore swollen vesicle included Spraguellatus species, Spraguelas fumigatus, Philevas, Terras, Niger, and this group make, as you see in this picture, vesicle at the end, and the conidia, it is a source. Conidia is a source of infection and help us for identification, and a source of reproduction, source of infection, and also reproductions or multiplication, and also it is one of the guideline for identification. As you see this in direct microscope, as we see by, if we isolate it, microorganisms Spraguelas, and see, we'll see this Spraguelas heads as black brown, and we see a fragment of conidia which help for reproduction, and this is the conidiophore, that means which carrier the vesicles. And from species to species, we have more than 150 species. It helps us for differentiate between them. We have conidiophore branching into penicillus. Penicillus is penicillium species. We have Bacillomyces, we have scleropsis. All these produce branching spiragallas. As you see here is conidiophore, one of the highly septated and the ways of reproduction. Also, we have Alternaria, similar like a sickle cell. As you see, there are many clusters of this part responsible for differentiated many types of this. We have Contridium conidiophore in a cluster. There are many species can produce in a cluster. We have a Creminium, Trichoderma, Glycolidium, and we have a Fusarium. This part of conidiophore always has been in a cluster. So this make colorful and beautiful. That means the fungi have many variable. So we concerning about the structure and reproduction to help us for identification. As we say that about the septated hyphoconidia born singly, like Cyriosporidium, we have Sidonium, and we have Schidosporium or Conosporium. We have Ebuspiremium, Pseudoloracea. This is the recent name classification. Ebuspiremium, that means Pseudoloracea boydii. All this genus, it can reproduce conidia only in the single form one. And this is exactly as you see, it is a single conidia. And the other part of the hyphae, according to their color, are very dark, dimeticulous. We have conidia in multicell. We call it alternaria. We have curvularia. We have drisillaria. Also, we have conidia single cell like Cladosporium. We have Nigrospora. We have Erosporodium. These are a part of these conidia. As you see, these conidias with multicellular conidia, and it have very dark color, as you see here also. This part of another species. We will speak about another part. This group are very familiar with aseptated hyphae. This we call it phycomycosis or zygomycosis group, according to classification. As we speak about Linnaeus classification of the according to the class and order and phycomycetes. So it included many species like Rhizobus, Mucor, Abbacidia, and Cunninghamella, Rhizomucor. As you see, we have many different types of sporanium, and this when they are mature rupture, and this is Abbacidia, and this Abbacidian connote many type of conidia. This by electronic microscope, you see columella here, and this is spore, and this is the ways of reproduction, and also this is a mucor. It have a sac, and this sac include sporanium, we call it, and inside it there is a spore, and this is sporanium 4, which is our carrier this part of and responsible. As you see, this electronic for septated hyphae. So morphological fungi, yeast, it is a different what about you speak about the different type of mold as septated and aseptated, other terminology coincident. We speak about dimorphic, mold, yeast, monomorphic, and dimorphic, according all this classification to morphology. And the mold classified to septated and aseptated, we speak about different classification according to reproduction, included with hyaline hyphae or dimeticulous hyphae. Now we will speak about the yeast. Yeast mainly it is similar like a bacteria in the morphology. Most of the when we grow these microorganisms are white creamy color, and most of them are no filamentous, unicellular, have a spherical oval in shape. Sometimes we find it as elongated, distributed in nature. Yeast sometimes are microflora in our body, as example like Candida or like Malassezia. It have a powdery coating or flutes or leaf, because yeast are colony of unicellular organisms produce as unit instead of colony, grow as a number of yeast, and this increase usually happen the ways of multiplication by budding. One cell of yeast can produce up to 24 data cell by budding. So some of the species yeast produce spot that fail to detach themselves, and this spot form short chain and this is called pseudo hyphae. It is not a true hyphae, but we call pseudo hyphae, and very presumptive for diagnosing direct specimen, especially in oral serage or vaginal infection. When we take a smearing from a sample by swab and we find this stage of pseudo hyphae, this very recognized microorganism indicated this invasive stage if the microorganism making pseudo hyphae. We have few types of the yeast produced by vision. Yeast are capable of facultative anaerobic, as we see in Saccharomyces. Some people use this in juice, and can lack of oxygen on organic compound as final electronic acceptor. It gives access to oxygen. Yeast perform aerobic respiration to metabolize carbohydrate to carbon dioxide and water and deny oxygen, and the yeast performed fermentation, because most of the fermentation of the yeast, it can be assimilated all the sugar carbohydrate and produce ethanol carbon dioxide, which base of brewing in baking industrial in a bakery. So this, as you see, this pseudo hyphae, it is the same as a sausage, and they are in above there is a budding of the yeast by gram stain, and other yeast shape we call it arthroconidia, like geotrichum and trichosporum, which are responsible of infection of certain type of the hair. As you see, this is a part of arthroconidia, as you see, Chiuritrichum, and also no hyphae present. No pseudohyphae can be produced by certain microorganisms, and this help for identification many types of the yeast, like Cryptococcus, like Rhodotella, Candida glabrata, and Saccharomyces. Most of this is Rhodotella, that means it have like orange color, always to be found in cold places as a contaminant. Cryptococcus, which are responsible for meningitis, we found in a dung of the pigeon. We have Candida glabrata and Saccharomyces. Most of them do not produce pseudohyphae. As you see, this Cryptococcus can produce capsule surrounded, as you see, in this part of this morphological. And also, the last one we want to speak about dimorphic fungi. We call it thermal dimorphism. That means changed according to temperature. No suitable condition can change it from tissue to culture to change part of morphological and also the part of this thing. Dimorphic fungi occur in two forms: as a yeast, as a mycelia, depending on environmental condition. In a parasitic or pathogenic form, fungi seen in tissue, exudated, or in a culture, a rich medium incubated at 2037. Dimorphism fungi are yeast. In a nature, when we grow at 25 centigrade free living, dimorphism fungi exist as mycelium. Conversion to the yeast from appear to be essential for pathogenicity of dimorphic fungi, and several medical important fungi are thermally dimorphism. These are very dangerous microbes of the fungi, lead to many complications, lead to death, like histoplasmosis, paracoccidioidomyces, blastomyces, and coccidioidomyces. This will lead to fatal, and also we have the sporotrichosis. All these parts of the fungi are dimorphism. In the tissue, we'll find it as a yeast. When we change the nutrition, not suitable media with rich like blood, like brain, like heart, so it cannot stimulate the growth similar like what we have in this part of this thing. So we classified our fungi into in according to morphological characterize, included also some reproduction ways into three ways. One is monomorphic, that if it is mold or yeast, and the third one is dimorphic, and that means the change according to their literature, and then we classified many different types. So our formative assessment in this part of this lecture to draw different type of hyphae according to this lecture, drawing because it help you for identification direct microscope, the shape of arthroconidia, septated hyphae, aseptated hyphae, and also budding yeast, and also different type of conidia, if it is multicellular, if it is in a conidia, also if it is in the single born, and also the branches of conidiophore, of example Sprigillus, and example of the ways of reproduction of aseptated hyphae of sporangio for the shape of this thing. I want every student to draw different type of the shape of different type of the hyphae, if it is hyaline, or if it is a dimeticulous, if it is in a cluster or a single born, of if it is a branches or no branches, if it is coincetic aseptated hyphae or septated hyphae, and different type of the yeast morphological to assessment from different type of the reference according to what we have say previously. If you return back to Adelaide University of Medical Mycology, or Dr. Fungus, or if you return back to PubMed or any website help you, then thank you very much for the second lecture. I hope you to benefit from this part, and thank you very much.